Good afternoon to everyone. Today I am going to talk about bacteria, its structure and characteristics. This is the content which cover whole topic of bacteria. Introduction, why we study about bacteria, occurrence and distribution, shape and size, structure of bacterial cell and characteristics. Now let us come into introduction. Bacteria are small single-celled prokaryotic organisms. Their cell structure is very simple. They are found in almost everywhere on Earth and play a vital role in the Earth's ecosystem. Many bacteria are living associated with animals including humans and plants. Some species can live under extreme conditions of temperature and pressure. Now the question is that why we study about bacteria? What are the interesting facts about the bacteria? Bacteria are directly related because bacteria are directly related to us by the points. It helps animals including human to digest food. They help trees by providing minerals available to them. Important, they also important in recycling of nutrients in the environment, and they used in biotechnology applications to produce everything from food to energy. They are also helpful to clean our environment by degrading wastes of soil, water and air. Now let us see the occurrence and distribution of bacteria. Where they found? Bacteria are ubiquitous in distribution. They are found everywhere in the world. The human body is full of bacteria and in fact is estimated to contain more bacterial cells than human cells. They may be beneficial or harmful to the other organisms. They can cause a number of dangerous diseases in plants, animals, including human beings. Some bacteria can survive under extreme environmental conditions such as hot springs, hypersaline environment and toxic environments, which could be lethal for any other life forms. Now let us see the shapes of bacteria. What are the shapes of bacteria? Various types of bacteria. These are some shapes. You know the coccus shape, Coccobacillus shape, Vibrio bacillus, spirochete and spirillum. You see in the picture that coccus is the spherical shape of bacteria. When the bacteria is elliptical, it is called Coccobacillus shape, and when it is appear like comma, it is called Vibrio shape. Like for example, some bacteria which cause disease cholera is Vibrio cholerae, and the bacillus bacteria is rod shaped bacteria. uh, Best example of the bacillus is lactobacillus, which occur in milk, which convert milk into curd. And the spiral shaped bacteria are called spirochete and spirillum. Now let us see the size of bacteria. The size of bacteria is in comparison to the eukaryotic cell is very small. In this diagram, in this figure, you see that the bacteria size of bacteria ranges from 1 micrometer to 1.5 micrometer. Now let us come to know the structure of bacterial cells. The bacterial cell is a prokaryotic cell, means it have incipient nucleus, what we call the nucleoid. The nucleoid is only nothing but only a DNA, not associated with histone and non-histone proteins, and the nuclear membrane is also absent in nucleoid. So the bacterial cells, we can divided the bacterial cell into two parts. The external uh, the structures external to the cell wall and the structures internal to the cell wall. The structures external to the cell wall are the peptidoglycan layer and flagella, cilia, and the fimbriae and pili. The glycocalyx layer is the outermost layer of the bacteria. It composed of polysaccharides and may contain proteins which protect cells and also help in adhesion. Flagella. Flagella are the long structures, whip-like structures, and baby filaments, appendages that produce from cell wall and are responsible for motility of the bacteria. This picture shows the electron microscopic diagram figure of the flagella. The flagella is divided into three parts: the basal body, hook, and the filament. The basal body of the filament is the most complex structure. The filament is long, hollow, rigid, cylindrical structure constructed of the protein flagellin. The filament is inserted into the curved tubular hook, and the hook is anchored to the cell by basal body. In gram-negative bacteria, the basal body have 4 rings, while in gram-positive bacteria, the basal body have only 2 rings. Now let us see the fimbriae and pili. These are also the external structures external to the cell wall. Fimbriae are small bristle-like structure sprouting out of the cell. It enables bacteria to attach solid surfaces. Pili are elongated tubular structures made up of a special protein called pilin. It facilitates attachment of bacterial cell to the host cell, are involved in the mating process. Now let us know about the bacterial cell wall. You know the bacterial cell wall is made up of peptidoglycan. But in eukaryotes, the cell wall is made up of cellulose. Now let us see in the figure. This is the yellow color layer is the peptidoglycan layer, which is cell wall, and by in increasing in size, but the magnifying image shows that it has the alternating units of N-acetylmuramic acid and N-acetylglucosamine. Now let us see the structures internal to the cell wall. Inside the cell, some components are found like cell membrane, mesosome, and nucleoid, which is called the incipient nucleus. The cell membrane is made up of phospholipid layer and proteins. It lies beneath the cell wall. It is 5 to 10 nanometer semi-permeable membrane which regulates the flow of material into and out of the cell. It is composed of, earlier I have talked about, it is composed of phospholipids and proteins. Now let us see, this is the mesosome. Mesosome is nothing but the enfolding of the cell membrane. Enfolding makes a structure which function like mitochondria in eukaryotic cells. In bacteria, it is used for energy production, like as mitochondria. Next, another main component of the internal structure of the bacterial cell is nucleoid. Nucleoid is the genetic material. Like in eukaryotic cells, chromosomes are genetic material, but here we are not saying it chromosome, because the DNA here is not associated with histones and non-histone proteins, and it is not enclosed in nuclear membrane, but lie only in the cytoplasm. Let us now Let us now see the another component, plasmid. Plasmid is the extra chromosomal genetic material which is only found in bacteria and any prokaryotic cells. Plasmid is a small circular double-stranded DNA molecule that is distinct from cell chromosome DNA. Plasmids naturally exist in bacterial cells. And another component is the ribosome. In bacteria and another prokaryotic cells, the ribosome is of 70S type, while in eukaryotes the ribosome is found in 80S type. On the basis of Gram staining, a type of staining, the whole group of bacteria can be divided into two groups: gram positive bacteria and gram negative bacteria. Gram is a stain. So now let us see the structure of cell wall of the bacteria. This is the cellular structure, the wall structure of the bacteria, which only differentiate into two forms, gram positive and gram negative bacteria, because gram positive bacteria have you see in the figure thicker thicker cell wall, and in gram negative bacteria the peptidoglycan layer or cell wall is very thin, due to which, due to the thicker and rough nature of the cell wall, it 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 contains the gram stain crystal violet. The crystal violet particles are adsorbed to the surface of the gram positive bacteria, while due to presence of slime layer or capsule in gram negative bacteria, the crystal violet is not adsorbed, and after washing with alcohol, it washed out, and then when we counter stain with safranin, the gram negative bacteria attain pink color stain, but gram positive bacteria not attain this stain. So by the staining procedure, we can easily divide it into two groups, gram positive and gram negative bacteria. This slide showed the major difference of gram positive and gram negative bacteria, which is structural differences and functional differences. Structural differences earlier I have discussed in the due to structure of the cell wall and the slime layer in gram positive and gram negative bacteria, and the functional differences are here, like lysozyme sensitivity. Gram positive bacteria are very sensitive, and gram negative are not largely resistant, not sensitive. Antibiotic permeability, very permeable. Gram positive bacteria and gram negative bacteria are not permeable. Sporulation in some species in gram positive bacteria, and sporulation not occur in gram negative bacteria. Exotoxin production, exotoxin production in some species of gram positive bacteria, and also in some species of gram negative bacteria. Then now overall we can conclude that the gram positive bacteria is more sensitive to antibiotics. So the diseases, we can say that diseases caused by gram positive bacteria are easily cured with the use of antibiotics, but the gram negative bacteria are difficult to cure by antibiotics due to presence of slime layer or capsule in the slime, an extra layer in the gram negative bacteria. So this is overall my presentation. Thank you very much. Thank you so much.